Hello everyone, I'm Alicia. I'm going to teach you about rolling circle of replication and inhibitors of replication. So I'll be talking about what is replication, then about rolling circle replication and inhibitors of replication. So first, what is DNA replication? Now, DNA replication, it is a biological process. It takes place in all cells during the cell cycle, that is during the S phase of the cell cycle. Now, what happens during DNA replication is that a DNA, it copies itself and produces two identical daughter DNA strands. And it is passed on to the daughter cells. So the replication, it takes place in a semi-conservative process. It means that one of the DNA strands will be from the parental strands and the second DNA strands will be the newly formed daughter strand. So it is always semi-conservative. The DNA strand is conserved. Now, the basic steps of replication are first is initiation, elongation, and termination. So this is a few short idea about what is DNA replication. Now next, we'll talk our topic, that is rolling circle replication. It is a type of DNA replication model. Now in rolling, it is mainly found in circular DNA or RNA molecule. Now, this method is mainly found in bacterial mating, in which a single linear DNA or RNA, it is transferred from the donor cell to the recipient cell. Now, this transfer from the donor to the recipient is usually through the rolling circle replication mechanism. Now, this mechanism, as I said, is mainly found in the circular DNA and plasmid DNA. Now, the rolling circle replication, it takes place in a unidirectional method. And this method or this model can produce multiple copies of single-stranded DNA and double-stranded DNAs. Now, as I said, the rolling circle replication, it takes place mainly in prokaryotes, that is the bacteria, and sometimes in eukaryotic viruses. Now, rolling circle replication, it is also called sigma replication. So what is sigma replication? During this model, at an intermediate stage, a structure is formed where it looks like a sigma. Hence, it is also called as sigma replication. Next, we'll talk about the enzymes that are involved in rolling circle replication. So the first enzyme is Rep-A protein. It is a regulatory protein. Now the Rep-A protein, it consists of, it is a dimer, okay? It is made up of two monomers and it contains active sites that has a tyrosine group. Now this Rep-A, it is a very important enzyme and it helps in the smooth functioning of the overall processes in the rolling circle replication. Now next enzyme is DNA polymerase 3. Now this is an enzyme that is mainly involved in the replication or the elongation stage for the formation of the new strand. Next enzyme, helicase. Helicase enzyme functions to unbind the DNA strand to open it so that the DNA polymerase 3 can come and bind and start the replication process. The last is a ligase enzyme. It is required, the main function is to join the gaps that are formed in the very end. So these are the enzymes Rep-A, protein, DNA polymerase 3, helicase, and ligase. Now, we'll start with the different stages or steps involved in rolling circle replication. Now, step 1 is initiation. Now, initiation, it starts by an initiator protein. It is formed by a covalent extension, which is why and starts the rolling circle replication. So what happens is that a circle DNA, as you can see on the right side, it has two sides, a DSO and a SSO. DSO stands for double strand origin of replication. I repeat, DSO stands for double strand origin of replication, and SSO stands for single strand origin of replication. Now, what happens during the initiation stage is that first, the rep A protein, as you can see, two red circles, the rep A protein, it goes and binds to the DSO of the circular DNA, double-stranded DNA. So once the rep A protein binds, it forms a nick on the DNA strand. Now, with the nick form, the triosine group, as I said earlier, the rep A protein has a triosine group. So triosine group, it remains attached to the 5' phosphate of the strand and thus forming the lagging strand. And the 3' strand acts as a leading strand. Also, the 3', it acts itself as a primer for the next processes. So the rep A protein binds, remains bound to the 5' end, forming the lagging strand, and the 3' end hydroxyl end, it forms the leading strand and also as the primer. So these are the basic steps involved during the initiation, the binding of rep A protein. Now, step 2, that is elongation. What happens during step 2 is that the rep A protein, it recruits the DNA polymerase 3 enzyme, which comes and binds to the origin of replication, all right? And the rep A also recruits a gene encoded helicase, that is PCRA helicase. The PCRA helicase, it unwinds the DNA for the replication process. Now the DNA polymerase, it continues the replication process in a rolling circle manner. That hence we get the name rolling circle mode of replication. Now the DNA polymerase in the replication process, it moves in a clockwise manner. So we can see in this image the helicase, it has unwound the DNA strand. The rep A protein that is remain bound in the 5' end of the single strand DNA that is being formed, and the DNA polymerase, this DNA polymerase, it helps the replication process. Now once all this is done, next and the last stage is termination. Now what happens during termination is that the replication has come to an end. It has reached the termination point. So the rep A protein, it joins the gap between the 5' and the 3' of the newly formed single strand DNA and separates itself from the old double-stranded DNA. So it forms a join, the rep A protein, and separates the single-stranded DNA from the double-stranded DNA. Now, there was also a gap in the double-stranded DNA. That gap is joined by the DNA ligase enzyme. And now this step, all these processes can be repeated many number of times to form single-stranded copies of DNA. So these are the various stages of rolling circle replication. But is this complete? No, there's a next stage. What happens over here is the single-stranded DNA that was previously formed, it forms a loop-like structure. As you can see over here, a loop-like structure is formed, and to this loop, it gives access to the RNA polymerase. The RNA polymerase comes and binds and acts as a primer. By acting as a primer, the post-DNA polymerase, it continues the replication process. As you can see here, it has continued the replication process and thereby forming another double-stranded DNA. So from this single strand, with the help of the RNA polymerase and DNA polymerase, we got another double-stranded DNA. And this comes to an end with the second cycle of replication of the single-stranded DNA. Now this explains what I've explained earlier. So rep A protein, it comes and it binds to the DSO of the double-stranded DNA. Once it binds, it forms a nick. As you can see, the rep A remains bound to the 5' end of the double-stranded DNA, and the 3' end hydroxyl group, it acts as a primer. Now, DNA polymerase 3 comes and binds to the ORI, and the Rep-A, as I said earlier, it recruits a gene-encoded helicase. Now, the helicase, it functions by unwinding the DNA so that the replication procedure can be continued. Now, the single-stranded binding protein is also present, and this continues the replication process. And in the end, we get, as I said earlier, one single-stranded DNA and a double-stranded DNA. This is the final result of replication circle. Now moving on to my next topic, that is inhibitors of replication. Inhibitors of replication, there are various drugs that are used to inhibit the replication process and thereby inhibit the cell division. Now this inhibitors, they can be used as antibacterial drugs, antiviral drugs, or anti-cancerous drugs, and hence they have many benefits and can be used for treating patients. So substances or examples of inhibitors are, first is quinolones. They are basically antibiotics. Their basic function is they inhibit the functioning of topoisomerase. By inhibiting the function of topoisomerase, it inhibits the action of DNA gyrase, and thereby it results in a DNA strand with many breaks. So the commonly available or the commercially available quinolones are norfloxacin and afloxacin. Now, the quinolones, they're also called as floxacin. So as I said, the topoisomerase 2, it is responsible, its function is to remove supercoils. So by the action of quinolones, this function is inhibited and thereby replication is stopped. Next is ciprofloxacin. This is another drug and it interferes with the DNA breakage and rejoining process. Ciplastin is another drug, the next. It is anti-cancer drug. It is used against many cancers like lung cancer, brain tumor, breast cancer, cervical cancer, etc. So how it does this is by it forms a intralink or a cross link between a purine base and the cisplatin. So a link is formed between the cisplatin and the purine, and thereby it prevents replication. So it is stopped. It cannot go for the next process. The next drug used is etoposide and adriamycin. These two drugs, it inhibits the mammalian topoisomerase. So the function of topoisomerase, as I said, is to remove supercoils. Okay, so that function will be inhibited by these two drugs. So etoposide and adriamycin, it is used as a cancer drug. And the last is novobiocin and nalidixic acid. These two drugs, they inhibit the enzymatic activity of DNA gyrase. So these are few examples of drugs that are used for inhibition of replication. And thereby, these drugs, they can stop the pathogenicity of bacteria and virus and can be a great boon for the humanity. These are my references. Thank you for listening. If you have any doubts, please ask.